Good morning, everybody. I think we should get started. Thanks to all of you for coming to this ceremony, the first ceremony of the day. So the most important one, I think, with some science. So we're very pleased today to have Ulrich Müller coming to receive his award. So I just want to remind you a few things about the foundation, what it is. Maybe not all of you are aware of it. So we do different things. We give research grants, but also some awards. Was the grand prize, and we're happy that we also last year's recipient, Barbara Canlon, that's also here. So hello, Barbara. So as you know, with the prize as you might know, with the prize now comes also a possibility for the recipient to do a sabbatical in France and to spend time in some French labs. And Barbara was the first guinea pig of the project. And it worked very well. I think you enjoyed it. So she came to Paris. I know she's in Montpellier. So, and I hope that Uli will do the same. So this is really, I think, a good opportunity also for French people to get to meet the prize recipient. So we just listed here the former recipient of the prize. There are also some prize from early career scientists, young investigator. Unfortunately, this year we don't have any application. So which is something I don't really understand. So we had so it's something that keep in mind that it's a very interesting prize. There's also money with it, and like for clinical and etc. we didn't have applications or applications that were not fitting with the call. So we're going to try to advertise even better, but we communicate with your young colleagues that they should really apply. So we hope we're going to get more applications in the future. So Ulrich Müller, so I guess many of you already know him. So Ulrich studied in Germany, in Cologne, and then he did most of his PhD in Princeton in the US. Then came back to Europe, to Basel at the Friedrich Schmiescher, and then went back to the US, to the West Coast, to work at the Scripps Institute until like 8 years ago, when he moved to the East Coast, closer to Europe, to Johns Hopkins University. So, I mean, I've known him for quite some time, but surprisingly not for his work initially on hearing. I knew him for the work he was doing. We were working on the same topic at some point, like cerebellum development, granular migration, and also cortex development. And that's how we met at a meeting a long time ago. But I guess you know more him, and I also know his work on hearing that he's going to talk about today. And Uli really had made some very important discoveries in the field of hearing, and also how the tip link is organized and how the air cells function. And so I guess, I mean, you really are very familiar with his work that he's going to present today. So congratulations again, Uli, and we're happy to have you here, and we hope you're going to come back next year or the year after to spend more time with us. So thanks a lot.
Okay, well, it's really a great honor to be here today, and it's the first time for me to visit the Fondation pour l'Audition, and I can see already, I know the community, of course, numerous of the researchers, and being here and seeing the building, being in France is really a treat, and I'm sure to take advantage of the opportunity that comes with this prize that Barbara already did to come and visit France and spend some time here. And I'm really very honored to be here, and it's really, really wonderful, and I'm very thankful for this. What I want to start my presentation with is to introduce you to the laboratory and also to bring to you an early connection to France. So my team, that is a team that works with me right now, and you see in the middle a colorful tree, and that tree is there because the group is as colorful as the tree. And there's one common trait between them. These people all have never worked on hearing research before they come to my laboratory. That's actually something very common throughout my career. I don't think I've ever managed to recruit anybody to my lab who had worked on hearing before, which I think in many ways turned out to be an advantage because the people bring all kinds of expertise to the field and make us look at the problems perhaps in a different way. I also want to share a story here from my early days where I was actually a researcher at the Friedrich Schmiescher Institute, and I was working on the cortex. And we made the strange observation that we had a mouse which was circling and rotating. We thought it's a cerebellar defect, but it turned out to be an inner ear defect. So we wrote up a paper. And I was a bit nervous because I knew nothing about the ear. So my colleague Denis Monard said, hey, why don't you go to Paris to Christine and get some advice? And I called up Christine, and she was so kind to invite me. And I came to Paris to the Pasteur, had a very nice day. Christine was generous with her time because there came this young guy who knew nothing. And she let me talk to all the people in lab. We published our first paper, and she encouraged us to stay in the field. And here we are, 20 years later, still in the field. What my laboratory fundamentally has been interested in the ear is mechanical sensing. Now, our body is full of mechanosensors, and for various modalities, perception of touch, posture and stretch, blood pressure, but also perception and sound and balance. And when we started working in the field, nothing really was known about the molecular machines underlying this various sensory phenomena. Each of them depends on mechanically gated ion channels, but what the ion channels were wasn't clear. Now, laboratory, which was initiated by Ardem Patapushian, established that the piezo channels are responsible for almost all mechanical senses, with the exception of sound and balance. Cezanne Wu in my lab, when he was in my lab, studied piezos in the ear and showed that actually nothing to do with sensation of sound. So the question really, the quest became, what are these sensors? And what fascinated us at the time about these sensors is that they're somewhat different. Actually, the inner ear mechanochannels are the fastest ion channels on the planet. They're activated in microseconds, while it takes milliseconds to activate other ion channels, such as the piezo channels. So the channel is somewhat fundamentally different. And also hearing is a really, really special organ. And as I'm sure most of you, perhaps all of you know, we can hear a vast frequency spectrum between humans, roughly between 20 and 20,000 hertz, whales up to 150,000, some dolphins even higher frequencies. We have an incredible discrimination power. We can sense frequency difference of about 0.2%. We have an enormous dynamic range, meaning we can hear how a needle drops on a glass plate or how an airplane roars over us, and we can distinguish that without damaging the sense organ. And this power spectrum is a trillion-fold difference between these two modalities that I just mentioned. And what the inner ear does, it really shows minute nanometer deflections of the sensory machinery to perceive these mechanical signals, the pressure waves, really sound pressure waves that we see. And as I'm sure you know, all of this is carried out in the inner ear, in this little organ here, this snail-shaped cochlea. And this cochlea, when you unwind this, is really built like a piano, where the low notes are, or the high notes are presented at one end, the low notes at the other end with a gradient in between, really quite like the keys on the piano. When one cuts a cross-section through this organ of corti, one sees the organ of corti here, which is really the sensory epithelium. And as most of you probably know, there are two types of sensory cells, the outer hair cells and the inner hair cells. And they have distinct functions, where the outer hair cells are really amplifiers. They amplify vibrations in the organ, which increases the sensitivity and frequency selectivity of our ear, while the inner hair cells send the signals back to the nervous system. What you see here is actually one of the first scanning EMs my first postdoc took from the inner ear and false colored. What you see here is the sensory cells at the surface. You see the stereocilia bundles, which have given these cells the name here, the 3 rows of inner hair cells and the 1 row of outer hair cells. And these stereocilia bundle are the business end of the hair cells. These are the mechanically sensitive organelles. And I'm showing this here in somewhat more detail, where you see this hair bundle here in a mammalian hair cell, where you have different rows of stereocilia, which are formed in this graded heights. And this polarity in the bundle is crucial for the function, because deflection in the direction of the longest stereocilia opens ion channel in the bundle, deflection in the opposite direction closes them. And I mentioned here 3 people who have been very instrumental to determine the properties of these bundles. Now, in 1984, a key discovery was made by Jim Pickles. He discovered this little filament here, which is called the tip link, which you cannot see in this preparation, but you see the electron-dense plugs, and this filament connects here this larger stereocilium to the shorter one. You see here's a larger and here's a shorter one. And the model was developed that this tip link conveys tension force on the transduction channels. Now, that was in 1984. And then we're about 20 years where it really was hard to solve this problem. What is the molecular underpinning of this mechanotransduction machinery? And one of the problems really has been in the field that the inner ear is encrusted in the temporal bone, which is the hardest bone in the body. It's as hard as elephant tusk. And these sensory cells in an inner ear of a human, we have 15,000. When you look at the retina, there are millions of photoreceptors, but having very few cells posed challenges. And then what we thought at the time, well, maybe we can use a trick that has been used in science a lot to solve unsolvable problems, or seemingly unsolvable problems. We turned to genetics. And as you all know, I have to mention Christine again here. A pioneer in the field was Christine, who actually is one of the first people, actually perhaps the first person, to positionally clone a gene linked to hearing loss. Now, and afterwards discovered many more, some of which I will mention. Now, what is really important if you want to do genetic screen, that you have a rich genetics. And deafness seemed to fit the bill because it's the most common form of sensory impairment in humans. And it's thought that roughly 500 genes are involved, or mutations in 500 genes. By now, mutations in over 150 genes have been identified. And what is really important for the research my lab and others have conducted, that there's nearly 100% conservation in the genetic landscape of deafness between mice and humans, allowing us to use mice as a model system to study the disease. And when we started to do then genetics in mice, at the time there were only 4 genes positionally cloned, which were linked to hearing loss. We didn't even have the mouse genome yet. But the human genome had just been finished, and the mouse genome was coming online. So there were parts of it done, but not the whole genome, but we knew it's coming. And what we applied is then a trick to the study of the auditory sense organ that had been used in other organisms. We carried out genetic screens. We randomly mutagenized mice, and then we screened them for hearing phenotypes. Actually, what brought me to America from Switzerland was the screen, because the Scripps Research Institute was willing to put up the money and facility to conduct the screen. So I took this gamble and left a comfortable position in Switzerland to go to a complete soft money position in America. But what we did there, we screened 2,000 families of mice that we generated, roughly 30,000 mice, and we identified about 30 gene mutations that linked to deafness. Other research has also identified gene mutations, and Christine as a leader and other, they identified many human genes that are linked to hearing loss, and mouse models became available for this mutation, or we mimicked them and generated them. And we have by now analyzed Weissman's mutation in over 70 genes. And you see here one of our early really screening pipelines, an early picture from Martin Schwann and the lab who carried out the screen. And Martin was a Latin dancer. Initially, when you clone the genes, you don't know what, when you identify the mice, you don't know what mutation they have. So he gave Latin dance names to the mice because they frequently also circle. And that was an analogy to one of the early mouse mutants, the waltzer mouse, which is also famous mouse with a hearing defect. We positionally cloned the genes. All of the mice were deaf, as shown here by this auditory brainstem recording. Some had vestibular defects, meaning balance was affected, and all were linked by us or others to human disease. So reinforcing the concept that I brought up before, that mice and humans have a very good convergence. Now, we then had to figure out which of these genes actually have something to do with mechanotransduction. And for this, we modified our screen. Once we had deaf mutants, we used electrophysiology. And what we do is we patch clamp onto hair cells, and we stimulate the hair bundle with a stiff glass probe. And then you can record. And I'm showing you these recordings here because you will see more of them. So when you deflect the hair bundle, the whole hair bundle with all its tip links, you open all the ion channels and get a large current. Or when you just push on it a little bit, you get a small current. This is tracing from a wild type and this from a mutant mouse. And you can quantitatively show this in the so-called current displacement plots, that as more you deflect the bundle in a wild type, as more current you get. And in a mutant, you may not do so because the transduction machinery is defective in some way. That is what a real screen looked like. That's actually one of our first ones. We have a number of these mice. We didn't even know yet what the genes are, and found one in particular which had defects. These are all quantitative evaluation of the data, but the traces tell the story, I think. Now, then, through the study of these mouse mutants involving a painstaking process, we identified 17 genes by now, which are linked in some way to the mechanotransduction process. And the first genes that we annotated were genes which have to do with the tip-link complex. And it was not just our work, this involved the work of other people, which I will in part mention here. So what we showed is this tip-link filament is consisting of a homodimer of cadherin 23, a cell adhesion molecule, which binds and trans to protocadherin 15, the second cell adhesion molecule. And at the upper end of the tip link, there's a little complex assembled, which consists of these 3 proteins here, all of them identified and linked to deafness by Christine. I can't avoid mentioning here. She has contributed so much. And but it turned out that this is the outline of this machinery. And I give you just 1 or 2 pieces of evidence supporting that. What you see here is an immunolocalization study where we stain the hair bundle for the actin in the bundle in red and for this cadherin 23 protein in green. And what you can see, it is not expressed at the very tip of the stereocell. It's not present here. It's further down. And that we showed with electron microscopy together with Bishara Kashar forms this upper part of the tip-link filament. And we did similar studies for the other proteins. We also got some structural information. Initially with negative staining electron microscopy, we could make out when we purified or expressed heterologously these proteins, they formed these filaments here shown for cadherin 23 and for protocadherin 15. At the time, there was no cryo-EM, but we started in more recent times to work on this. A few years ago, we got a low-resolution structure which showed already this cadherin repeats, this repeat domains here shown by these dots. And more recently, not published yet, we actually got a cryo-EM structure of the whole filament where we can see each of the repeat units that forms this filament and where they touch each other, shown here for the protocadherin 15 filament. And what is really intriguing, of course, we and others can align this with mutations along this filament. And I think this is really remarkable. We went from positional cloning, not we identified protocadherin 15 first, others did, but the identification of this mutation which got deafness, we go down to the molecular mechanisms now, how these mutations actually cause hearing loss. We can precisely ask how these different mutations affect the function of this protein to get a really molecular understanding of the pathogenesis of the disease. Now, one thing that you notice with this picture, I mentioned at the beginning, there must be an ion channel which conducts the ions, which is really guarded or controlled by the activity, by the pulling at the side of this tip link. And in 2009, a very influential paper came up by Robert Fetty-Place and Tony Ritchie, which suggested that the ion channel must be at the lower end of the tip link. They didn't know what the ion channel is, but the ion channel passes calcium ions. So they imaged the calcium entry site and showed at the lower end of the tip link, suggesting really the striking functional asymmetry in this filament with an ion channel at the lower end and a tension machinery, as we think, at the upper end. Now then, having genetic initially didn't help us to find components of the ion channel, but we took advantage of the fact that it at the lower end, shown here blown up, and asked, well, can we identify proteins that bind to protocadherin 15? And Bo Zhao in my laboratory, he identified this protein TMIE, which he showed can bind to the tip link filament. And what was interesting about this again, TMIE had already been identified as a gene had been identified in human patients to be linked to hearing loss. While we didn't get it from our genetic screens, they're never complete, there was a link for deafness already. In parallel studies, Jeff Hall's laboratory showed that there's another protein, it's actually two, they're called TMC1 and 2. They're also localized in stereocilia, and Teresa Nicholson and Peter Bar-Gillespie showed that they can also bind to protocartier 15, suggesting then, well, we have proteins which sit down there at the tip links, have transmembrane domains. Could there be ion channels? They're localized in the stereocilia bundle. And what Chris in my lab showed, these two proteins actually can also form a complex with each other. They don't just bind to protocartier 15. You can see a TMC1 and its close homologue, TMC2, can bind to TMIE. When you generate mutant mice, we did this for TMIE, Jeff Holt and Andy Griffith did this for a TMC, you see there's no mechanotransduction current. You have here the current in a wild type and here in the knockout mice. Now, this in itself is all very promising, but it doesn't say these are ion channel components. And here the field for a while hit a roadblock. We and others tried to express these proteins in heterologous cells to show they truly form ion channels. And that was a real failure until today. Because the TMC protein in particular, when you express it in heterologous cells, gets stuck in the ER, so you can't really record from it. But you can still analyze proteins like this and ask if they're a component of ion channels by working in hair cells. And that is by making mutations and asking, do they change in vivo the properties which are intrinsic to the channel? And we did that, and Jeff did this for TMIE, and Jeff did it for TMC, and you all, I think, know Jeff several years ago, rightfully also got an award for his really tremendous work on TMC1. What Jeff showed that point mutation in TMC1, 2 can change the conductance and ion selectivity of the channel, which are two intrinsic properties of the channel, also supported by similar work carried out by Robert Fattyplace. And we did the same for TMIE. And I will just show you some examples of our work from this. And what came once again to our help was the genetics, because there were 3 mutations, point mutation, linked to deafness in human patients which affected the TMIE molecule. Now, we used, with the advent of CRISPR-Cas technology, it became pretty easy for us to put these mutations into the mouse genome and ask, what does it actually do to TMIE? And what you see here is, again, electrophysiological recordings from a control where you have nice currents. But when we put in, shown you for one of these point mutations, when we recorded from them, currents were much reduced, as you can see here too. We showed that some TMC proteins are still in the bundle, in the wild type and in the mutant. Tip links are there too. We did all these controls. There's some loss of TMC proteins. I'm jumping here a little bit, so some of the reduction in current has to do with loss of TMCs, but there's plenty of TMC there to analyze what it's doing. And what we wanted to ask, are the properties of ion channels changed? Now keep in mind, when we record from the hair bundles, we usually record all the channels at all the tip links, but there's a trick we can apply. We can puff EGTA on this bundle, and these filaments here are formed by caterin standing for calcium-dependent adhesion molecules, so we can break these tip links in a live hair cell until we have one tip link left. And we can patch on the cell body of the cells and record mechanotransduction current from a single channel then. And that's what these recordings look like. What you see here are controls. So you held a bundle, then you deflect it, and the ion channel pops open. And from these traces, you can determine ion channel properties, such as the conductance, how many ions flow through the channel. You should see here from a mutant, its channel seems smaller, the size of the current, and you can see this here, the conductance in the mutant is changed, which suggests an intrinsic property of the channel is changed. We can also ask questions about the ion selectivity, and to do that, you determine the reversal potential of the channel. We did that. Again, it was changed in the mutant. So intrinsic properties of the channel are changed by these point mutations linked to human deafness, suggesting then that this transduction channel complex is actually made of this unusual configuration of the TMC protein together with TMIE. Now, then before this recent revolution with AlphaFold, still several laboratories started to predict the structure of these TMC and TMIE proteins with more conventional tools, and predicted that TMC proteins actually form a dimeric channel complex consisting of two proteins, each with certain transmembrane domains, with the TMIE protein linked in some way to this complex. So these were structural predictions. And many people, including us, tried and still trying to solve the structure of the mammalian complex, and we are coming to that. Now, one other protein was shown to be really important, mechanotransduction, and this is a CIB2 protein that stands for calcium and integrin binding protein 2. Actually, some of this work was done here in the laboratory of Aziz together with Christine, where they showed that mutations in CIB2 affect actually mechanotransduction. Now, Xiaoping Yang in my laboratory solved the true structure of this protein. That was the true structure we got of one of the components linked to mechanotransduction. What he also showed that CIB2 binds to this little domain here of TMC1 that was not clearly predicted in the original structure prediction. But this, he solved actually the crystal structure of CIB2 in the complex with this fragment of TMC1. And this gave us one very important and interesting clue. And that is the CIB2 protein looks very similar to KChIP1 or to calmodulin. What I've highlighted to you here is some alpha helices of these proteins, and you see the similar arrangement of this protein. So what these showed that CIB2 belongs actually to a family which is known as the neuronal calcium sensors. Calmodulin looks very similar. And what is truly remarkable, so these proteins regulate voltage-gated potassium channel and mechanically gated channel. There's some evolutionary theme here. And you see here a space filling diagram of this TMC, and I have animated this for you. And I guess I have to go out of the laser pointer mode, otherwise the movie won't play. And you see here the protein turning around, and you see up there this loop domain. It's actually helix domain, which finds in the little pocket. And the binding specificity in this pocket differs between different of this calcium sensor protein, and that determines the ion channel specificity. So this basic configuration is the same. They have calcium binding domains down here, but this pocket varies between the proteins, giving specificity for the ion channels. Now, this gave us then a model of the ion channel, which looks a little bit even more complex. And that is where we have now essentially 6 proteins seem to assemble into this ion channel complex. Now, as I said, we and others tried very, with a lot of effort to solve the structure and still working on this. But then we got help. And as it is in science, sometimes help comes from unusual places, in this case from the nematode TMC1. Now, TMC1 is expressed in nematodes, and a laboratory, Tang et al., they showed actually that TMC1 is required in touch neurons in the head of the worm to mediate touch responses. I have to switch out again of this mode to show this to you. What you see here, what this looks like actually, that is from Qiang Yang in my laboratory. He looked at this in more detail. When the worm hits an obstacle, it turns around and withdraws this direction of movement. And what Qiang recently showed, we just published this, is that this touch response depends on the C. elegans orthologs of these 3 proteins, which form the mechanically gated ion channel in mammals. Now, Eric Kuhl, who is a structural biologist, he took millions of worms. And then he purified TMC1 from the worms. And what he purified, actually very surprising, is at the time, is TMC1 in complex with CIB2 and TMIE. And Eric solved the crystal structure as a cryo-EM structure of this complex. And that's what it looks like. This is now a true structure. And what I find really remarkable, the complex, the way they isolated and determined then, it was predicted essentially from the work from us and others, from the biochemistry and genetics and mammals. And actually, even the binding domain down here for CIB2, he now got this helices edge, just like the helices we had actually crystallized from the mammalian protein. Now, on the ion channel conductance pathway, some work from Jeff and Robert suggested here, although I think there's still substantial room to think about the ion pathway, and that's something we're working on. Now, this then has led to a model of the mechanotransduction channel complex. And that is that we have here this extraordinary molecular machine with a molecular asymmetry and a highly complex ion channel, which consists of far more proteins than initially appreciated. Now, the next question that we want to take on, that's very recent work, and I'll show you also a number of things which are not published yet, is how is this ion channel actually gated by force? What is the mechanism for channel gating? Now, there's two models which have been proposed for gating mechanically gated ion channel. One is this lipid stretch model, where the lipid bilayer is deformed and opens the ion channel. The other is where the ion channel is tethered extracellular and intracellularly, and deflections against this tether point open the channel. Now, the tip link suggests that there's a tether process, but it isn't clear if the tip link is directly linked or if it could deflect the membrane and pull on the membrane in some ways. The lipid layer model, here's a protein, seem to be gated by this, while the tether model applied to NUMP-C. But which one is true for this complex? And that's a question we took on next, because nothing in this channel complex inherently tells you how you get mechanical sensitivity. So what is the mechanism of gating? Now, what came here to help for us is again genetic. And actually this little molecule here, which has a strange name, I apologize for this, we didn't give these names to the genes. I will refer it to L5. This is actually the first gene we identified from our screen and linked to mechanotransduction, the first we published, and one of the last we are starting to understand. So this protein has 4 transmembrane domains. And what we showed already Wei Zhang in the lab and in this study, that it binds to protocatenin 15 and to TMC1. It is also localized in the tip link region, which you see here, and I have to again switch back to the laser pointer, which is, as you see here, this is the tip link region, which is where this short and longer stereocilia meet. Now, one thing gave us a clue that this protein may have something to do with mechanical gating. When you study mutant mice, which don't have LHFPL5 or L5, you see transduction currents are much reduced. That has in part to do that the transduction machinery falls apart. But the currents that are left, you can analyze. And I'm showing you here a blow up. You see here a current from wild type and mutant superimposed. It was a relatively long time course for this ion channel. When we blow up just this initial phase here, we see how the channel gets activated. Activation of the wild type channel is shown here, and it's so fast, we actually don't know the actual time constant because our probes don't push hard enough, fast enough. But you can see that the mutant is severely delaying this activation. That's shown here more quantitatively. The activation time constant of the wild type is very fast, and this is so delayed that we can measure it. That suggested to us there's something wrong with force transfer onto the channel. So suggesting then when this here is our ion channel complex, well, perhaps this L5 molecule helps to link this complex together and transfer force onto this channel. Now, is that true? We started with how could it link to the channel? Now, where could it anchor actually to this tippling complex? And we started looking at the nematode complex. And when you look at it, what you can see here in the center of the molecule where the two TMC molecules move, there seems to be a groove. In the meantime, AlphaFold, this amazing tool, came online, and we could actually predict channel structure for the mammalian channel. And that's what the mammalian channel looks like by this prediction. A little different. In particular, we get no information on the extracellular and intracellular domains, which differ somewhat from the nematode channel. But what is remarkable, using the supercomputer facility at Hopkins, and a very talented graduate student in the laboratory who's a computer scientist and was only a mathematics and computer science background, we actually assembled the transduction complex together with the tip link, and it took the computer several days. But what we showed, that near this TMC10 domains, sorry, in the center, the tip link, the highest probability structure predicted by the models the tip link right into here, where we have the protocardian 15 dimer and the L5 dimer sitting in the center. I'm showing you a view now from the top from this complex, where we can see sort of the transmembrane helices. So what you see here is the TMC molecules and the transmembrane helices. And here are these little grooves, and that's exactly where this protein would fit in. What we also like very much, you see here these arms, which are specific for the mammalian protein. They align with this domain of protocardian 15. This does not exist in the nematode channel, and nematodes don't have tip links. So there seems to be something very special here. I tell you also, we now have some deafness mutation in this domain. We're working on this. Now, but this view also gives you some hints to help me explain to you the experiments we did. So what you see here are the transmembrane domains 10 from TMC1. Next to it sits this L5 and protocardian 15 molecules. So we ask now, do these molecules truly bind to each other? What is the spatial configuration when we use biochemistry? And for this, nature helped us actually. So this is TMC1 molecule shown here with its 10 transmembrane domain in slightly different form, which is easier to visualize what I want to visualize. And there's a close homologue, TMC3, which looks a little bit different, but it's very closely related. We had actually shown that TM3 cannot substitute for TM1 for promote mechanotransductions in hair cells, something I'm not going to show you. But having these two proteins, we could now ask, well, do they both bind to the tip link? And the answer is no. TMC1 does, but TMC3 does not. So this is very useful. Now we can make chimera and express these proteins and map domains which are important to bind to other components of the complex, such as L5. And I'm showing you an example for this. So what we did actually, and I'm showing you only one, and that is a successful chimera, of course, where we took the TM10 domain from TMC1 and planted on TMC3, also one from TMC3, and we planted it on TMC1. And then we asked, what happens to the binding activity? Can this domain here confer binding, which our structural prediction suggests? And that is what happened. TMC1 does not bind, TM3 does not bind, but this molecule nicely binds now, showing that transmembrane domain 10 is really necessary for binding. When we have the opposite chimera, there is still binding left because we know other domains also contribute to binding. But what is crucial here is that transmembrane domain is crucial for binding, just consistent with the structural model we had developed. Now, there's a second prediction, and that is that these helices here, TM1, transmembrane domain 1, from L5, also binds to these domains. That's what the structure predicted. And nature was helpful to us too. For L5, there's a homologue, L3, and this homologue, L3, we could use to make chimera with L5 protein with different domains. And we were very lucky also because we had shown L3 cannot bind to TMC. So what that's shown here, L5 can bind now to the TMC3 construct with the transmembrane domain 10 from TMC1. L3 doesn't bind. But when we transplant these two helices on TMC3, we regain binding activity, which again is predicted by the model. This helix 1 here is predicted to mediate interaction. And we have made more mutations like this, but they all show really we can find out precisely what binds and what not. This one here gives us an additional tool. We can ask, is binding actually crucial for function? And the way we did this is shown here now, because we had developed this little trick. Now we can make little organ explants from the inner ear, for example, from an L5 mutant mouse. When you record transduction currents here, there's very little here. But now we can stick these different proteins in there and ask, can they rescue transduction? We would predict that proteins that bind rescue, while others that don't bind don't rescue transduction. And that is actually what worked out and is shown here. When we stick L5 into the stereocilia, here you see the hair cells without L5, there's no transduction. We stick in L5, not surprisingly, we can reconstitute transduction. But when we stick in this molecule, we can recover this transduction, but not with this. So there's a nice conversion of binding and function. And we have substantiated with a number of other mutants, which I'm not going to discuss here. But all of this is really consistent with the model that this L5 molecule binds in the center of the TMC molecule to serve as a force coupler to the tip link. I did not show you all the binding data to protocartier 15, but we have similar data for protocartier 15. But I think the point has come across, I hope, how this complex links directly. We also like this model very much because we have two ion channels here, which are linked to this dimer of TMC1 CIB2. And in the center, pulling on it may ensure all of the two pores get equal force distribution and are opened with a similar kinetics with a very fast time constant, which is a prerequisite for the function of this channel. Now, also far so good. We had these data 5 years ago, but we only published them recently. And that has to do that we hit one roadblock with one mutation which didn't fit that model quite, or we didn't understand it. And we had made one chimera between L5 and L3, which didn't fit the bill. And this chimera, we had just taken this little cytoplasmic domain from L3 and transplanted it on L5. And we gave it N-terminus L3 on L5, we named it. The only difference is the 3 amino acids which are lacking in L3 in an isoleucine to leucine substitution. Now, we expressed this in here. We showed that this little mutation does not affect binding. Binding is very nicely maintained when we make this mutation. But when we tested function, this mutation does not allow for function. So even though this protein here very nicely binds through the tip link protocartier 15, binds to TMC through this transmembrane domain, it cannot activate the ion channel. We get no activity. And really, we could not figure out what's going on. We started to worry. And I have to give great credit to Chu-Feng Qiu, who did this work, that for years he worked on this, first in the dark, because we didn't have structure. But when structural information came up, things fell into more pattern. It's the last story I really want to tell you. Now, one thing which has been observed, to study that, Chu-Feng put this mutation into the mouse. He used CRISPR to swap this little cytotail from L3 into L5. When he made the mutant mouse and analyzed it, he showed all the proteins, protocartier 15, L5, TMC, they go to the hair bundle, just like the wild-type proteins. The whole transduction machinery gets assembled in the stereocilia bundle. Proteins are there. When he recorded transduction currents, they were severely reduced. There was reduced mechanotransduction, even though all the components are there. Then he carried out single channel recordings. There were normal single channel recordings. What this suggests that we have the channel in the bundle, they just can't get activated. But how can that be? Everything seems to be there, it binds together, but we can't activate the channel. Now, what helped us really to solve this problem was structural biology. Now, one thing which has been observed for all mechanically gated ion channels, that they have something very peculiar, which is called amphipathic alpha helices. So these helices have a hydrophilic side and a hydrophobic side, and they can make curvature in the membrane. When you look in the channel that is the bacterial mechanotransduction channel, MSCL, one of the first that was identified, you can see this amphipathic helixes just right at the interface between the membrane and the cytoplasm. And these domains are crucial for response to mechanical force. Another example shown here is the NOMP-C channel, which has a transmembrane, this huge anchoring repeats, which link it to the cytoskeleton. You see here, again, this amphipathic helices, which has an interface of the membrane with the cytoplasm. Again, these domains are crucial for mechanical gating. And this goes for all mechanical gated ion channels that we know. They all have these domains. Now, we started to look in the TMC protein complex. And what we observed, lo and behold, there are amphipathic helixes. This amphipathic helix here is actually from the TMC1 molecule. And it is called H3 because next to it, two other helixes, which are called H1 and H2, which are not amphipathic. This other helix here is from TMIE, and that is, we showed H1 as it's called, and it's also amphipathic. So they all have amphipathic character. And when you spin around these molecules, you see again they sit close to the membrane interface. Actually, this is slightly, I aligned this slightly wrong with the membrane here. So they sit right in the position between the membrane and the cytoplasm. This helix from TMIE is actually longer, as predicted by AlphaFold. It's just that short from the crystals, from the cryo-EM structure. It's available. Now, one other observation then we made from our structural prediction is that the tippling protein sits right in here, and this little N-terminus from L5 sits right next to this amphipathic helix in TMC1. So this little helix, which doesn't affect binding, which is crucial for function, is very close to this amphipathic helix. So we wondered if it maybe has a function to gate the channel through this helix. And the first question we asked, is this helix, which actually sits here in the N-terminus of the channel and is preceded by these two helix, is this helix important for mechanotransduction? Again, this time we took this organ explant from TMC1 mutants and stuck in different TMC constructs, and that is shown here. TMC hair cells don't have mechanotransduction. You stick in TMC1, you can recover it. A mutation in TMC3 abolishes activation of the channel, even though it's in the bundle. Suggesting this is alpha helix is truly important for channel function. Now, the next question we ask is this channel, does this interact with L5? So what we ask then here, if we take these proteins, can they bind this domain? And does a mutation affect binding? And that is true. And you can see this. L5, the wild type, can bind. L3 binding to the amphipathic helix is drastically reduced. One other observation we made has this activation of the channel. Mutation, this mutation here I had shown you severely reduces but does not abolish mechanotransduction. Now, when we look at the remaining transducer current, we can say that they are normalized in a certain way. We have to, for these things, we normalize to total current, but what that reveals is the mechanical force sensitivity. The channel sensitivity in this mutant of the remaining channels is shifted to the right, meaning there's a problem in force transfer onto the channel. Now, these studies then together suggest a model for channel gating, how these L5 molecules actually work. And that is what we think in the model we are working on, that this mechanotransduction channel, that L5 may serve as a molecular coupler, while this TMIE molecule may serve as a molecular gatekeeper. And how we envision it is that the mechanical force which pulls on the channel is transferred through H3 on this ion channel, leading to a conformational change which helps open the pore. And perhaps even this gate swings open to change conductance. And we think this is the case because the mutations in TMIE, which affect conductance, sitting in this domain here and not in the transmembrane domain. It's not part of the pore pathway. Conductance is changed by some other path, and we think by gating the channel. And that is a model we're working right now to really prove this in detail. But so far, it's consistent with all the data that we have. Now, the link to the cytoskeleton, which a lot of these channels which are tethered, like here through the pivot, is unclear. A candidate for this is the IB2 molecule, something we are working on. But there are other candidates, I can tell you. One candidate which has actually been suggested from nematodes are the anchorins. There are 3 anchorins in mammals. We have made triple conditional knockout. The paper should come out soon. Anchorins in mammals are not required for mechanotransduction. It's different in nematodes from mammals. Now, I think we have from 1984, when the tippling filament was discovered, come a long way where we know what seemed to be a simple problem, a simple protein forming the tippling and a simple protein forming the transduction channel is really a very complex molecular machine. And I think what's remarkable, mutations in each of these genes is linked to hearing loss in humans. And what this really tells us is the power of combination of many laboratories to get a molecular understanding of the pathogenesis of a disease, where we have a class of diseases which we perhaps can characterize or classify as mechanotransduction diseases. And I'm finished with an inside story from the laboratory. And we have some other genes I'm not talking about. And that has to do with the platypus. And it may surprise you that I'm talking about a platypus. But the platypus is really a fancy organism. It was introduced from Australia to British science in the late 1800s. And when the researchers brought it to England, people thought that's a joke. Some anatomists had stitched this together. And the reason is it's a mammal and it lays eggs. This is unusual. It does electrolocation and has venomous spurs. That's venoms that belong to spiders and snakes, not to mammals. A duck bill, a beaver tail, an otter-like web feet. Well, we refer actually in the laboratory as a transduction at a molecular platypus. And why is that? Well, in this machinery, different parts which are used elsewhere in the body have been reconfigured. Caterians usually form adherence junctions, but here's a very special caterians which forms these tipplings that have been reconfigured. TMC1 protein and its homologue are related to lipid scramblase, which regulate lipid properties of membranes. CIB2 is related to this sensory subunits from voltage-gated potassium channels. L5 has homology to top proteins which regulate glutamate receptor activity. We have amphipathic helices, which are found in all this transduction channel. And what I've not written down, we have TMIE, which is unique, and we don't have anything else anywhere else in the body. But this machinery really looks sort of like a platypus, where it takes these different elements, sticks them together to make this wonderful molecular machinery, which has kept us busy for the last 20 years. And with this, I want to thank my laboratory from our Christmas party from last year, many collaborators which have contributed one or the other way, former lab members which are not on this photo, which have gone to bigger and better things, running their labs now in different other places. And I also would invite you to Baltimore. That's in a harbor. Johns Hopkins is somewhere in the back. It's just a short drive to the harbor. Please come and visit us whenever you have an opportunity. And with this, I thank you for your attention.